Oh, hi. The objective of this lesson, we will apply modern terms and concepts to explain the work of Gregor Mendel. First, to the terms. Gene, a portion of DNA that eventually leads to a trait. So we know that the truth really is that genes are a portion of DNA, and that DNA leads to a protein, and it's the proteins that lead to the traits. Now, you get two of each type, one from mom, one from dad. Allele, an alternative form of a gene. We often talk about eye color as a trait. And with that, we talk about the genes that lead to it. We talk about blue eyes versus brown eyes. And if you recall from years past in science, we recognize those as letters, maybe big B for the brown eyes and little b for the blue eyes. More on that later. Back to the terms. Dominant gene, the form of a gene that, when present, will show its trait regardless of the other gene it is paired with. Recessive gene, the form of a gene that, when present, needs to be paired with another recessive gene to show its trait. So, if combined with a dominant allele for the same trait, its present is silent. It's kind of like here is that trait for that recessive gene, but the dominant one will come and cover it over and you won't even see it. It is silent. So, this organism is said to be a carrier because the recessive trait can be passed on to the offspring of that organism. Phenotype, the observable trait that results from the combination of the two alleles. An example, we can see brown eyes. Genotype is the genes themselves. An example, for the above situation, well, for brown eyes, is it big B, big B? Well, we know that this is for brown eyes, that's for brown eyes. So, of course, we're going to see brown eyes, but we could also have big B, little b. Here we have the trait for brown eyes. Here's for blue eyes, but that's recessive, so we don't even see that. Right? Homozygous dominant. The genotype is two dominant alleles. So with the example that we've been using, it's big B, big B. Homozygous recessive. The genotype is two recessive alleles. So that would be big B, little b, little b. And heterozygous. The genotype is one dominant, so that's a big B, and typo, and one recessive, a little b. So now we get to the story. This is Gregor Mendel. He lived in the 1800s, kind of like when Darwin lived. You know, we had Darwin's work. Darwin's work got strengthened by our understanding of genetics. And this guy is kind of like the father of understanding heredity. He was an Austrian monk, and he investigated pea plants and how they passed on their traits to other offspring. He noticed that when pea plants self pollinated, they make virtual clones of each other. But when they cross pollinated, the traits change. So I think we need to know a little bit about what self pollination versus cross pollination means. And we actually need to know a little bit about a flower itself. So plants. Plants have both sets of genes come from one source. So that means that the plant is both a male and a female at the same time. It has male parts and female parts. And here they are, pollen grain. That is the male sex cell of a plant, while the ovary, in essence, eggs, let's call it, is the female part. And if the pollen ends up meeting the egg, the ovary of the female, just of the plant itself. So if the male cells that are produced, the male sex cells that are produced by this plant meet the female sex cells, well, genes didn't come from any place else except for itself. It's going to make a clone. So it's going to self pollinate. And that's what was happening with many of his plants. And he noticed that nothing was changing. But there is also another option. If, let's call this plant one and plant two, if the pollen of one ends up migrating by way of a bee to plant two, we now have the genes from plant one meeting the genes from plant two. We're going to get cross pollination. And then that dominant and recessive issue comes into play, and traits might change. So, can the passage of traits from one generation to another be predicted? He looked to make observations by manipulating the variables. He could actually choose which pollen and which ovaries fertilized each other. So he was controlling an experiment. He took what he called true breeders that displayed a certain trait, such as purple flowers, and crossed them with another, such as white flowers. We now know that these true breeders were homozygous for their traits. They had the same allele. So this purple flower had big R, had the dominant genes, and the white flower had r, the recessive. He then observed the traits of the offspring, what he called the F1 generation, and he then took the offspring and self pollinated to produce an F2 generation. Let's use a tool that we've seen before called a Punnett square and dig deeper into this. This is a modern tool applied to his work. So, what happens when true breeders for the same allele self pollinate or even cross pollinate? Well, here let's identify this as the male and this the female. You'll see why when I show you the Punnett square. And when they self pollinate, we end up with 100% of the offspring resemble the parents. And it could work like this. So here is the first one, and that's the male symbol. So the two options to pass on to the offspring are big R, and well, you can only pass on one to the offspring. So let's put that one here and the other one here. Now, this other plant, we'll identify it as the female. Big R, and the same thing. The egg can have either a big R or a big R. That's all that this one can pass on. So, what happens when this pollen grain, is in essence, the sperm of the plant, meets the egg? Well, big R, homozygous dominant. What happens when this one meets that one? Again, big R, and the same for the other two. In this case, all offspring are homozygous. On to another option. So, what happens when true breeders that have different alleles are crossed? So, here we have the homozygous dominant crossing with the homozygous recessive. And what was observed was 100% of them were purple. That is the phenotype, that is the observable trait. But what were the actual genes? So again, let's identify this one as the male, this one as the female, so we could use the Punnett square. Up top, we have big R. The only options to pass on to the child, the offspring, are big R's. So this pollen grain has a big R, so does this one. In this case here, the female, the only thing those eggs, those ovums, those ovaries are going to have are the little r's. What happens when they cross? Big R, little r, that's our heterozygous. And so we see that the genotype is big R, little r, but the phenotype is purple. And that's the same with the other three situations. 100% of the offspring are purple. All right. But we know that there's a recessive trait in there. He didn't, but he found it by checking what would happen if the offspring of this last example, well, those children grow up and have their own children. So we have the heterozygote crossing with a heterozygote. Again, let's identify this as the male, this as the female, and look at the scenario. So it is a heterozygote. One pollen grain will have a big R, while the other option a little. The female is also a heterozygote, so one ovary with the big R, one with the little. What happens when that pollen meets that ovary? Big R, big R. We end up with the genotype homozygous dominant and the phenotype purple. If this pollen meets that ovary, we have big R, little r, our heterozygote situation. Our genotype is big R, little r. Our phenotype is purple. What happens if that pollen meets that ovary? Again, another heterozygote. The phenotype is purple. But what happens if this pollen grain meets that ovary? Little r, little r. Are homozygous, recessive. And now the purple, I'm sorry, the white returned. Hmm. Let's look at the math because Mendel was big on math. He noticed that one quarter of them, I'm sorry, he noticed that three quarters were purple. That's all he truly knew about it. And he noticed that one quarter were white. Now we have that here. Our one quarter are our homozygous recessive. But of that three quarters, actually one half of them were heterozygous, which means we had the big R, little r, and one quarter of this three quarters was homozygous dominant. So big R, big R. And that's how our modern understanding applies to some of his work. So here is where the magic lies. Just a recap. We had a purple flowered pea plant with a white. The offspring showed purple, as though the white never existed. But when the offspring had offspring of their own, the white reappeared. So this is in the way. We'll just go through it like this. Where the white flowers disappeared, in later generations, they reappeared. That kind of summarizes the magic of Mendel. But his work didn't stop there. He applied this method with other traits of pea plants, not just the flower color, but flower position, seed color, seed shape, pod size, pod color, stem length, and so forth. And he crossed them, and consistently he got the same results. And we can now use it to predict the outcome of selective breeding. So, if we were to take a Labrador retriever and mix it with a standard poodle, we end up with a Labrador, a fantastic seeing eye dog. So it has the loyalty and the ability to learn and be obedient like a Labrador retriever and be hypoallergenic so that people that are blind don't have an issue with being allergic to their own dog. So, what would happen if we crossed a dog with another species, such as human? All right, that can't happen. I wish I were showing you this on April Fool's because that can't happen because the chromosome numbers of the different species are not the same, and offspring would never even develop. So, we now know that Mendel, back to that guy, he actually got extremely lucky because all the traits he studied were determined by one gene. So one gene led to one protein, led to one trait. They were kind of isolated in that fashion. He got very lucky because that's not reality. The fact is, most traits are controlled by the combination of more than one gene. And it kind of looks like this. If these are three genes per chance, it will make three proteins, and the combination of those proteins actually make the trait happen. Further, sometimes one gene plays a role in many traits, and that looks like this. So one gene, that one protein that the gene makes, intermingles with many other things and leads to trait A, trait B, trait C, and many more, in fact. And we also know that when we have different traits, sometimes they blend together. And instead of just seeing one masking the other. It's a blend of both of them. And this Punnett square below represents that, where we have a light colored flower mixed with a dark, in this case, white and red, producing pink. So we now know, again, that he got lucky. Or shall I say, we got lucky. Because if it weren't for the insights we learned from Gregor Mendel, we may not have gone further in understanding things in the way that we did, in the timeline that we did. I mean, we would have eventually gotten there, but not as fast without this man. So I hope that this helps